I welcome on behalf of Indian Society of Weed Science and Directorate of Weed Research. First of all, I welcome all of you. I welcome Dr. S.K. Chaudhary, Deputy Director General, ICR, Dr. A.K. Pandey Sahab, who is Chairman as well as Vice Chancellor of Vikram University, Ujjain. I welcome Dr. S. Bhaskar Sahab, ADG, Indian Council of Agriculture Research, New Delhi. I welcome on the dais Dr. J.S. Mishra, who is Director, ICR, Directorate of Weed Research. And I welcome again Dr. Steve, who has joined this meeting from Australia. And I see galaxy of the weed scientists like Dr. Gogoi, Dr. Yaduraju Saab, Dr. Samundar Singh, and many other weed scientists like Dr. Chinnamuthu and dear ISWS members and friends. You know that today we are going to organize this webinar 8 under the series of webinars being conducted jointly by Directorate of Weed Research and Indian Society of Weed Science. First of all, it is customary to light of the lamps. So I will request Mr. Gyanendra for the lighting of the lamp by the honorable guest. Our this Saraswati College with the symbol of knowledge. Let us play the ICR song, please.
जय जय कृषि परिषद भारत की सुखद प्रतीक हरित भारत की कृषि धन पशुधन मानव जीवन दुग्ध मत्स्य फल यंत्र सुवर्धन वैज्ञानिक विधि नव तकनीके पारिस्थितिकी का संरक्षण शस्य श्यामला छवि भारत की जय जय कृषि परिषद भारत की। हिम प्रदेश से सागर तट तक मरुधरती से पूर्वोत्तर तक हर पथ पर है मित्र कृषक की शिक्षा शोध प्रसार सकल तक आशा स्वावलंबित भारत की जय जय कृषि परिषद भारत की जय जय कृषि परिषद भारत की।
Thank you. Now I invite Dr. J.S. Mishra, Director ICAR-DWR, for setting the context.
Unmute.
Dr. J. Mishra Sahab, please unmute yourself.
Nice.
Okay. Yeah. Dr. Mishra Sahab, I invite you for setting the context, please.
Thank you. Thank you very much, Dr. Sushil Kumar sir. A very good morning to all my senior colleagues, Honorable DDG Sahab, ADG Sahab, all Honorable Vice Chancellors and all senior colleagues, senior dignitaries, directors of various ICR institutes, and the guest speaker of the day, Dr. Steve Adkins, and all my senior colleagues and friends. As we all know that the Parthenium is really a big challenge. It is a global problem, and it has become one of the rampant weed throughout the globe. causing enormous damage to the agriculture productivity, human and animal health, as well as the biodiversity. And in India, this weed has assumed a serious concern, not only in non-cropped area, but now it has become a major weed of the cropped area, as well as the city dwellings, roadsides, and all institutional campuses and villages and city areas. And we know that the Directorate of Weed Research is functioning since last more than 33 years. And this institute has developed the Parthenium management technology as well as its utilization. But the very important thing is this weed cannot be managed by a single management techniques. So the awareness, to create awareness among the stakeholders along with the management, it is a very important aspect, because until and unless we create awareness, it is very difficult to eliminate this weed from the society. So keeping in view the importance of this weed, the institute, the ICR is celebrating Parthenium Awareness Week since last 16 years. It started from 2015. In fact, it was the thinking of the then director, Dr. Yadraju Saab, who is very much there in the audience, started this program. And by this date, it has become a mega program of the ICR, and every stakeholder throughout the country, we are creating the awareness. And moreover, this year, this program is very important because we are commemorating the 75th year of the India's independence and giving the name of Azadi Ka Amrit Mahotsav. And that's why this Parthenium Week, we are celebrating this year in a grand manner. And for this purpose, we have invited Dr. Steve Adkins from the University of Queensland. Of course, he is delivering his lecture after a few minutes. And added to this, in this week, we have published more than 10,000 posters and printing materials. We have sent through the mails, through the speed post to all stakeholders, may it be KBKs or universities or directorates, ICR institutions, or even the municipality, Gram Panchayats, state government organization, just to create the awareness of the ill effect of the Parthenium. And in this way, we have also planned to organize the different types of webinars, training programs, as well as the press conferences, so that more and more stakeholders should know that in what way we can eliminate the Parthenium, or how we can utilize the Parthenium for vermicomposting and things like that. So with this brief remark, I once again thank you very much and inviting all colleagues to join this program. And Honorable DG Saab and Secretary there also has given his appeal, and that which we have published in the posters which we have circulated throughout the country for eliminating the Parthenium from the society. So with this brief remark, now I will request Dr. Sushil Kumar to go for further proceedings. Dr. Kumar.
Thank you very much, Dr. Mishra Saab, for setting the context and giving a brief introduction of Parthenium Awareness Week. With us today, as a guest of honor, our beloved Dr. S. Bhaskar is with us. Dr. Bhaskar is Assistant Director General in ICR. Now I request Dr. Bhaskar Sahab for his address. Dr. Bhaskar Sahab.
Good morning, everybody. Honorable DDG NRM and Chief Guest of the today's program, Dr. S.K. Chaudhary. Of course, he is attending some parallel program with Minister. He is not able to participate now. I think he might have given his recorded message also. Dr. A.K. Pandey, Vice Chancellor, Vikram University, Ujjain, and Chairman of the today's program. Vice Chancellors of different state agriculture institutes who are joining this program today. Dr. Steve Adkins, Professor, University of Queensland, Australia, and lead speaker of the talk. Director colleagues, of different ICR institutes. Dr. N.T. Eduraju, former director of Directorate of Weed Research and chairman of Research Advisory Committee of the Institute. And many other stalwarts in the field of weed science have also joined. My director colleague, Dr. J.S. Mishra ji, Directorate of Weed Research, and scientists, students, friends, and the main pillar of this program, Dr. Sushil Kumar. He is also the president of Indian Society of Weed Science. Dr. Samudra Singh is there. Dr. Katreshan is there. Chinnamuthu is there from this society also. If you see the Parthenium and its history in the country, people say that it has traveled from other countries, alien wheat, through our PL 480 program during 1960s or 70s, when India was facing a lot of food shortage problems. There was food deficits. We used to import, and Public Law 480 program of USAID. So along with wheat, it has come to a place called Kadakwasala in Pune, and first started from the railway tracks, and gradually it has entered to almost all the places, cropped as well as uncropped, and the civic amenity sites, and the all other aspects. You see wherever you today in India, you can see Parthenium much more than any other thing. So that caused a lot of damage to agriculture productivity, human and animal health, as well as our agrobiodiversity has also lost because of the invasion of this alien weed into our agriculture system. Of course, that is a kind of prevention we talk. We could not prevent it because of so many reasons. And Dr. Steve Adkins is here, and Australia is in a very strictly prohibited or prevented this weed entry into the Australia, it seems. They are successful in doing that, but we are not. So it has assumed serious proportions of losses, and it has not left even the campuses, whatever may be the campus, college, university, or administrative buildings, even if you go to the collector or district magistrate offices in the country, you see Parthenium first. Parthenium is there to invite all the guests compared to any other person. That is the kind of situation we have created ourselves. And how to eliminate is a question now. How to eliminate from the system is a moot point for the country like India, because we have failed in our duties for preventing this weed into the country. Now only we have to eliminate, or we have to control or contain this weed, but at least in the agriculture fields, so that it should not intervene in the agriculture production. That is a thing we have to give a lot of emphasis today. So ICAR Directorate of Weed Research, Jabalpur, has developed very effective technologies for its management and utilization and all those things. Whatever possible, what is possible, what is not possible, it is again a questionable mark. Because I told already prevention is, we have lost the opportunity of preventing it. Then I am also very happy with the Institute's efforts, which organizes Parthenium Awareness Week, because we realize today that it is not the development of technologies or the disseminating the technologies, but more and more, since it has invaded all the places, not only agriculture, non-agriculture places and all those things, we need to create a awareness in the minds of the public or all the stakeholders today, because the entire 130 crore population of India are the stakeholders for this, as far as the animal health and that. It is called by different names by different countries, or common names are there. In entire North India, it is called gajar grass. Probably the process stage of the Parthenium, it looks like gajar. That's all. Once it grows, it is totally different from the gajar. It is, but in South India, it is called as Congress grass. I don't know why this name called. Maybe because of the people say that the inflorescence looks like cap of congressman. No congressman wears cap today. So I think we don't need to call this again anymore Congress grass. So we can call it as a gajar grass or Parthenium itself. Parthenium is about, now it has become vernacular almost. Even a common man, the uneducated farmer also knows that it is called Parthenium only. So it universal name it has acquired because it is a universal weed. So today they are organizing 26th Parthenium awareness. That is being organized by this institute. And this week will go from today, 16th to 22nd August 2021 throughout the country. I appeal to all the stakeholders to participate in the activity as a component of Swachh Bharat Abhiyan. You see, yesterday also the Honorable Prime Minister, while addressing the nation on the eve of 75th Independence Day, he has taken this reference. And he wants to take this Swachh Bharat to the very height, the newer heights. So that is a kind of mission we are going to have as far as the Parthenium or Swachh Bharat Abhiyan is concerned. It is a very easily can be integrated. We can be integrated. It's very easily integrated with the Swachh Bharat Abhiyan. So we should take opportunity of this Swachh Bharat Fund, and you can talk to many corporates, the corporates also and corporations also, municipal corporations. I mean, municipal corporate means the CSR funds. CSR can also come from the industrialists for controlling of these things. So when you do that, when you make any campus as a free Parthenium campus, no doubt it is going to set the ecological standards and which will ultimately culminate into one health in the country. Now, one health, one nation is also talk of today. So we should strive towards ourselves for achieving this goal of one health, one nation also. The Parthenium management techniques, of course, the DWR has come out with several technologies, but uprooting of Parthenium at very young stage, it's very important thing. But few people maybe the health conscious, they may be allergic to the Parthenium also. But I don't know, see, at a very young stage, the germination stage, if you go for repeated harrowings itself, it can control. But people neglect at that stage. But repeated surface harrowing itself can take out many of the germinated Parthenium seedlings. So it reduces the seed bank in the soil also. So just like stale seed bed technique here, see when the monsoon is starting or beginning, I think after 15 days or within 28 days, if you go for a small harrowing, it can take care of the uprooting of many of Parthenium. We don't need to employ the laborers also now, because a lot of things are being mechanized now. The mechanization is quite spreading in India, and India is the largest country in exporting tractors now. When it is exporting tractors, why don't we do ourselves mechanization in the country? That is also one of the questions it is being there before the policymakers now. And of course, many fields are left fallows. This is, of course, this is not possible in the country like India, where we have we say that it is a freedom of the farmer to grow or leave the land fallow, or you may not lease because of the land reforms or the land laws have to come out. Just like farm laws, we should also think of some land farm laws also. Then only it is possible that no land should be left with fallow. So then we can take care of the the cultural, the mechanical control of the weeds, particularly in the fallow lands. The another thing is fast bulking crops. Are there any fast bulking crops which can grow much faster, having more vigorous growth, both in terms of shoot as well as fruit, so that it can suppress the Parthenium? At least one season in between. If you go for such fast bulking crops, one such example is maybe Susmania, Dianchak and goose, where you will get the window between two crops. So then to little extent the Parthenium infestation can be reduced. But whereas chemicals are concerned, of course, you have listed some chemicals and all those things. Glyphosate is already under it's a ban. I don't know what is going to happen. We are discussing at national level how to revoke that ban and all those things. And we are yet to see, but you have listed another thing, metribuzin and 2,4-D also, but a lot of things are coming now, organic farming, natural farming, and all those things. Under such situation, how you can use the chemicals is again a questionable mark. And biological control, of course, Dr. Sushil Kumar knows the limitation of biological control. And no doubt we have a very successful model of biological control, but the perpetuation and the continuous multiplication of the bioagents in a given location is more important. And not only during rainy season, beyond the rainy season is also required. And these are all some of the issues which should be discussed at length, and we should come out with some kind of integrated management and all those things. And of course, convergence, they can be easily converged as far as the elimination is concerned with Swachh Bharat Abhiyan or Manrega. Manrega can also be used in controlling this Parthenium in non-cropped lands, uncropped lands, and you can also use it for the other aspects also. As far as utilization is concerned, of course, the Parthenium, the composting that you are telling that, instead of, it is again a tedious practice. Instead of that, we can incorporate then and there itself. I told you in the rosa stage, if you go for a harrowing, little bit of harrowing, it can be incorporated then and there itself. And there you can apply some kind of, some integration is required. Some, you can think of the decomposing organisms. When you incorporate this Parthenium at a very young stage, it will be very, very soft and succulent also. It can be easily attacked by some of the organisms, microorganisms, which can decompose very fast in the field itself. That kind of innovation is also required. Of course, legal aspects, it is left to the policy makers. We cannot touch those aspects. And I am very happy to note that again, this Parthenium awareness, which is very much essential in country like India. We should take it to the nook and corner of the country. So we should educate the farmers, and we should share the knowledge wherever possible. Either single method or integrating 2 or 3 methods can be used to control this Parthenium. So I wish you all the best on this occasion. And of course, you will be doing your wonderful job today. And you have invited a intelligent professor and knowledgeable professor from Australia, who can talk, give, and much more information on this alien invasive weed, and it is causing a lot of problems for a country like India. Thank you very much. Thank you for giving me the opportunity.
Thank you very much, sir, for your nice talk and giving insight to us about the management of Parthenium in India. As now I will invite our chief guest address Dr. S.K. Chaudhary Sahab, as Dr. Bhaskar Sahab already told us that he is in a parallel meeting. So, but he was sincerely, he has given a message for all the audience of today's webinar. So I will request Mr. Gyanendra to play the recordings of Dr. our DDG Sahab.
Good morning, ladies and gentlemen. Wishing you all a very happy Independence Day, the 75th Independence Day, which has happened just yesterday. It is very, very important that this international webinar on the Parthenium weed problem and its management at the global level is being organized by our Weed Science Institute at Jabalpur. This is very much timely because we are organizing these events when India is celebrating its 75th Independence Day, and all the year will have variety of celebration under the theme Azadi Ka Amrit Mahotsav. And in today's function, Dr. A.K. Pandey ji, Vice Chancellor of Vikram University, Ujjain, and Chairman of this program, is also present. Vice Chancellors of different state agriculture universities are also present here. Dr. Steve Atkins, Professor from University of Queensland, Australia, will be giving a keynote talk today. Also, we have Dr. Suryanarayana Bhaskar, our Assistant Director General from the ICAR headquarters. My director colleagues from different ICAR institutes. I find Dr. Yaduraju, also the former director of DWR and present chairman of RSC. Sushil Kumar ji, the president of Indian Society of Weed Science, and other senior colleagues. I'm very happy to note that ICAR, the Directorate of Weed Research Jabalpur, is regularly organizing Parthenium Awareness Week since 2005. Means for the last 15-16 years, this program is continuously being implemented or being organized across the country. And a kind of awareness, public awareness, is also created on the ill effects of Parthenium and also motivate people for the management of Parthenium. Parthenium in India, it is commonly known as Gajar Ghas or carrot grass, and has now become a problematic global weed. It's originally regarded as a major weed in Australia and India. This weed is now widespread in around 48 countries in Africa, Asia, and South Pacific, and has the potential to spread to new regions in future. After noticeable occurrence of Parthenium in Pune, particularly Maharashtra, in 1956, it has spread like a wildfire across the country. Initially, this Parthenium was a problem in waste and vacant land only, but now it has become a problematic weed in cultivated fields as well. In forests, it has become a serious problem in grasslands, particularly in national parks, on which herbivorous, carnivorous food chain survives. It has been estimated that about 35 million hectare land are infested seriously with Parthenium problem in India. So out of 330 million hectare total geographical area, or out of 140 million hectare, if you consider this area, it is huge. The increase in Parthenium infestation in crop area in recent past is alarming. And many of the states, including the central government, has invested very heavily in management and removal of this Parthenium. Elimination or removal is completely not possible, but management is possible. That is what Government of India and different state governments, they are doing. This alien weed is believed to have been introduced into India as contaminants in PL 480 feed. When we imported that, Public Law 480 passed in 1954 to give food grains to developing countries for eliminating starvation and malnutrition that time. And PL 480 wheat was imported from USA in 1950s. While local spread has been achieved through multiple pathways, including roads and vehicles and all, by water, wind, and everything. So this was a problem which we imported or which we invited. So this is a very chronic problem now, because it started in mid-60s and it's continuing after so many years. Parthenium has immense tolerant ability against high and low temperatures and water salt stress, besides able to survive in adverse conditions. And this is why it is able to be successfully present across different agroecologies in India. It has allelopathic potential, producing a number of growth-inhibiting phytochemicals which can suppress the growth and germination of surrounding flora, causing immense losses to biodiversity. The excess reproductive ability to this weed, it facilitates its invasion within suitable habitats. So what is the impact? In general, if you see, Parthenium is a poisonous, pernicious, problematic allergy and aggressive weed, posing a serious threat to human being and livestock also. In India and Australia, this weed has been considered as one of the greatest source of dermatitis, asthma, nasal dermal, and nasobronchial type of diseases. So many people, they get allergy because of this. Besides all effects and efforts, it also causes several other problems like reduction in agricultural production up to 80% in some cases. Blockages of common pathways, reduces the aesthetic value of the parks, roads, the avenue plantation, gardens, and residential colonies. These impacts have become more apparent and prominent as the weeds invasion has expanded from less than 20 countries now to more than 40 countries at present, making it a serious invasive plant and one of the world's worst weed. And at present, we see that it has spread in 48 countries globally. How to manage it? There are so many options and so many steps also. The success of Parthenium management based on the integration of all the available techniques. No single technique stand alone will be sufficient. And integration of these techniques and its implementation with public participation is the only way out. It's not only that 1 week we celebrated Parthenium Awareness Week and the rest of the year, rest of the 51 week, we forget it. If you do so, then we will never be able to eliminate this weed from the world. Its immense ability to generate and produce seeds in every season really is a big threat. One method will never be successful. Therefore, we require integrated approach. We require legal management at the highest level. The state and central government should declare Parthenium as noxious weed and implement law to hold responsible the people who are responsible for spreading this. Preventive management, very, very effective method, but hardly implemented in India. However, in Australia, this method is strictly practiced, preventive management. Uprooting of Parthenium, the only way out. Now, there are several programs like Mahatma Gandhi, Narega, and so many other programs by which there are Swachhata Mohims or Swachhata Abhiyan, by which about many efforts are being made to uproot this noxious weed and provide hand gloves to people who are working with this and uprooting program, and should be advised to wear full slim shirts and all this precautionary measures are required. Also, we have some ways and means of mechanical management. In many crops, particularly fields, this Parthenium germinate profusely when left as fallow for 1 season or so. In such situation, mechanical deep plowing before flowering is good and economical. Now, there is a trade-off between one side we say conservation agriculture or zero tillage, the other side we recommend this. So there may be some problem, but in some areas, certainly this mechanical management will be an effective option. Also, the cultural operation, the fast-growing cover crop like Salvinia, or what we popularly call Dhecha, it suppresses the growth of Parthenium. Salvinia adds fertility to the field, the soil. Also, by use of chemicals, we can knock down Parthenium, but these are very, very costly, and these are the chemicals which will have their effect decades together or years together. Metribuzin, for example, 0.5 to 0.75 kg, it can be used as pre-emergence just after sowing to control that, you know very well. Also, we have some methods of biological control and also utilization of Parthenium. This can be used for various purposes like for biopesticide preparation, because having antifidant properties against some insects, enhancing feeding of silkworm on mulberry leaves by spraying leaf extract for making paper and composite. However, the most effective and cheap method to make compost of uprooted Parthenium by pit method developed by DWR, by which seeds can completely be cleaned. Therefore, management of this weed is very, very important priority for India, not only India, globally, this is a great priority now. And it requires collective and continuous effort at the community level, not single individual level or single institute level, and creating awareness time and again, adoption of integrated approaches to manage this noxious weed. This year, Parthenium Awareness Week is being organized by this directorate from tomorrow to 1 week throughout the country. And I request and I appeal to all the vice chancellors, not only those who have joined today, but to all the vice chancellors of state agriculture universities, all the directors of ICR institutes and in charges of KVKs to participate in this activity wholeheartedly and make it a component of Swachh Bharat Abhiyan and ensure Parthenium-free campus and also Parthenium-free agricultural fields wherever you are working, either your own fields or the farmers' fields where you are working in. So with this, I would like to stop here, and I wish you all the best for today's discussion, deliberation, and also this Parthenium awareness campaign and this Parthenium week will really provide a better solution for future, and it will create a great awareness in not only the country but across the globe. The message will go. And today we have our guest, we have a lead speaker. I wish all the best to this generation. Thank you very much. I will not be able to continue here because I have another meeting. So I will take leave now. So thank you very much to the organizers for inviting me for this important event. Thank you. Namaskar.
Thank you very much, sir. In fact, due to unavoidable circumstances, he had to go in other meeting, but we are really very much thankful to him that in spite of his very much busy schedule, he has given his message, and just now he is in other meeting. So now with us, Dr. A.K. Pandey. Dr. A.K. Pandey is chairman of this program also. And before I invite him for his address, I wish to introduce Dr. Pandey that before becoming the Vice Chancellor in Vikram University in Ujjain, Professor Pandey was here in Rani Durgavati Vishwavidyalaya, in Rani Durgavati University in Jabalpur, and he has done a lot of work on Parthenium. In fact, he has obtained his DSc degree also on the Parthenium, particularly integrated management. He is special in pathogens. So now, sir, I will invite you for your kind remarks. Dr. A.K. Pandey Sahab.
Thank you, Dr. Singh Sahab. On behalf of the Vikram University Ujjain and Maharishi Pandey Sanskrit Vishwavidyalaya, Sanskrit and Vedic Vishwavidyalaya Ujjain. Actually, I am heading that university also. On behalf of these two universities, I welcome to all of you associated with this seminar, especially Dr. Suresh Kumar ji, his Honorable DG ICAR, Dr. Bhaskar, Assistant DG, and one of our most respectable person who has already sometimes he was a director in RCW, and he played very important role in the initial establishment of this NRCW, Dr. Edu Raju, Dr. J.S. Mishra ji, Dr. Sushil Kumar, and one of our foreign guests, Dr. Steve Adkins from Australia, and most of the honorable vice chancellors of the Agriculture University. First of all, I would like to invite you for this darshan to Mahakal, this in Ujjain. Actually, Ujjain is all of you know this Ujjain is one of the most important pilgrims. This place in India, not only in India but in world also, people are coming to worship our this Jyotirling. One of the Jyotirling is here in Mahakal, and Ujjain is very famous. Ujjain actually earlier known as a Ujjaini, and when Nalanda and other Vishwavidyalaya started, the same times this Ujjaini, especially one of the ashram, Gurukul, Sandapani ashram, where our Lord Krishna, he had taken this education, entire education from this place. So I invite to all of you, and our university will provide all the hospitality, and even I would like to request Dr. J.S. Mishra to organize some of the program. This Indian Wheat Society has also sometimes visit to our place, and invite jointly we have to organize some of the program. You have heard our Director General and Assistant Director General, and they have given entire scenario of the Parthenium, but I would like to share few of the things. Parthenium is now, especially in the era of the COVID, people have reduced their immunity, and in this stage Parthenium may be very hazardous and may cause pandemic to very very large population will be affected if we properly not manage this Parthenium. Yeah. Parthenium, as our Director General, he has already told, he has discussed, give a elaborative view of Parthenium. Parthenium, as you know, they cause the dermatitis, contact dermatitis, even asthmatic peoples. And even there was a report in Madhya Pradesh where about more than 5 cough, they have died because of the consumption of this Parthenium. So Parthenium is very important and very dangerous, very challenging weed now in India, and almost encroaches all the ecosystem, all the habitats, human habiting area to agricultural land in forest area. So it has to be managed very effectively. And NRCW is doing very well. I have associated with the NRCW since its inception. So they are doing very well. And this awareness is one of the most important step. That is rather first step to manage the proper this weed. So they are doing very well. And this Pakhwada 1 week program definitely will provide a very insight in the human population for the management of this weed. I, again, because of the paucity of the time, I cannot just talk too much about this. We have to heard our foreign speaker, Dr. Edith Stephen. So I again invite to all of you people, and thanks to the people associated with this program. Jai Hind.
Thank you very much, sir, for your kind words. And certainly we will make the program on Parthenium at some point of time to your esteemed university. So before we start the next technical session, on behalf of the Directorate of Bead Research and Indian Society of Bead Science, let me thank Honorable DG sir for sending his message in form of appeal, which has been sent every corner of the country to make the people aware of the Parthenium. As we all heard, our respected DDG sir, in spite of his busy program, it shows his great concern that how much he are concerned with the Parthenium and with the institute, and he has given very nicely his talk as a chief guest talk on the occasion. Our chairman, Dr. A.K. Pandey Sahab, is already with us every time so far as the Parthenium management is concerned. So thank you very much, sir. Our guest of honor, ADG Sahab, Dr. S. Bhaskar Sahab, he has very precisely given the overall context of the Parthenium management in the country. So thank you very much, sir, for your gracious presence. All the directors of ICAR Institute, Honorable Vice Chancellors, Director Extension of the Rani Lakshmi Bai Agriculture, Central Agriculture University, Jhansi, Dr. S. Singh Sahab, ADG colleagues, Dr. J.P. Mishra, ADG of International Relations in ICAR, other ADG colleagues, our senior colleagues like Dr. N.T. Raju Sahab, Dr. A.K. Gogoi Sahab, Dr. Samundar Singh, President of International Bead Science Society, Dr. Kathiresan, and many senior learned colleagues, scientists, and friends. Thank you very much for joining this inaugural program and making it a success. And I hope that you all will be there in the technical program. So before inviting Professor Steve Adkins from the University of Queensland for his lead talk, just a few words about Professor Adkins. Professor Adkins is currently in the University of Queensland, Australia, and he did his PhD from the University of Reading, UK, and his postdoc from the Canada and Australia. He has a 33 years research experience on the Parthenium and other beads, particularly on the bead ecology and bead seed ecology. He has been the principal investigator of many projects worth rupees more than $15 million. And he has more than 180 publications to his credit. And even he has published good papers in the Nature, as well as in the proceedings of the National Academy of Sciences. He has supervised more than 60 postgraduate students and more than 40 other students on the various aspects of the bead management. And he is a founder member of the International Parthenium Bead Network, and he has been the twice president of the Asian Pacific Bead Science Society. So with this brief remark, we are really grateful to Dr. Steve for his presence on the occasion of this Parthenium Awareness Week. So with this brief remark, I invite Professor Adkins for his talk. So Professor Atkins, please.
Well, thank you very much for that wonderful introduction. I hope everybody can hear me. Is that correct?
Yes.
Yeah, excellent. So I'll just attempt to share my screen so we can look at my presentation. So is something coming up?
Yeah, it's there, sir.
Yes?
Yes.
Excellent. Well, it gives me great pleasure to be online to talk with you today at this very special occasion of your 75th Independence year. And of course, at the start of your 16th Parthenium Awareness Week. I think this is a wonderful activity that you run every year. It's a shame that more countries don't do the same. My country, like yours, has a major Parthenium problem, but unfortunately, not that very many people know about a particular weed. So what I'm going to present today is about the problems that this weed is causing, not only India, not only Australia, but around the globe, and the ways that other countries, as well as Australia and India, are attempting to manage this particular weed. But just to begin with, I want to let you know that I'm from the University of Queensland, which is in the city of Brisbane, which is in the state of Queensland in Australia, a very large state, probably about 2/3 of the size of India. And on my graph here, I have the area where Parthenium weed is a major problem shown in gold. So you can see the area over which the weed spreads is a very large region. So what I'd like to do is just take you through 4 different topics. First of all, I'd like to explain to you about the global situation of this weed, explain about the kinds of problems the weed is causing, then take a look at the different management approaches that are being used around the globe. Also take a look about what might occur in the future. I think we need to think about this as well. And finally end with a few closing remarks. So I think everybody knows what Parthenium weed looks like. It's a plant, of course, that normally completes its life cycle in under a year. We call it an annual plant, but in actual fact, there are a few circumstances it can grow into the 2nd year. It really depends on the environment. So the question really comes, why is this particular plant such a major problem around the globe? In over 60 different countries it's found in at the moment. Perhaps it's because there's no natural enemies that attack the plant once it arrives in a new country. Perhaps it's something to do with the unique physiology of the plant, being both a C3 and a C4 photosynthetic plant. Perhaps it's to do with its tolerances, its ability to tolerate many abiotic stresses, particularly pollution, hence the reason why it grows along the sides of the roads in amongst their cities. But perhaps it's also to do with the genetic variability that we find in this particular species, Parthenium histophorus. But it's almost certainly something to do with the unique reproductive system this plant has. And I think we've already heard that it's a plant that can produce 30, 40, perhaps even 50,000 seeds per plant. Some of those seeds have dormancy, which enable it to persist in the seed bank for many years. It will germinate under a wide range of temperatures, so the plant can appear virtually any time during the season when there is good rainfall. It does form these very large seed banks, and it will germinate even in the stressful environments that it's often found in. And as you can see from this particular photograph, the image on the far side there is an X-ray of the seeds coming from the plant. And as you can see, the vast majority are filled seeds and able to germinate. So not only does it produce a lot of seeds, many, if not all of these seeds can actually germinate at some stage. So we believe the plant originated from probably the Mexico region or the Caribbean. There is a few people that argue that maybe it came from the northern part of Argentina, Bolivia, Paraguay. And the reasons they say that is because there's a fair amount of genetic diversity to be found in that region as well as around Mexico. And in fact, the plants in that region do have a different anatomy and morphology and actually have yellow flowers. But it's very interesting that this form of the plant is never spread around the globe. It's always been the one that is found in the Mexico region, the one with the nice white flowers. So it's this particular form of the Parthenium plant that is spread around the globe. And you can see the number of countries that now have the Parthenium present. Some of these countries have been lucky enough to eradicate it, and some of them are only just in the process of getting the weed. This particular graph shows the spread of the weed around the globe. So in actual fact, the very first reports of it in another country were way back in the 1700s in Mauritius. How it got there, it's not clear, but probably the traders in the southern Indian Ocean brought it there. And in that early period of time, it spread to a number of countries in those regions. Then in the 1930s, it made an appearance in China and Vietnam. And also at the same time, over in Southern Africa, it made an appearance. But then perhaps the worst times that the weed invaded was in the 1950s, when both India and Australia received importation of, in our case, pasture seed lots for planting new pastures. And in India, a wheat lot probably brought in for food aid. And in amongst it, of course, was the seeds of this weed. And from that point on, as you can see from the graph, the number of countries that have become invaded is dramatically increased. So this time in the 1950s was a very important time for the spread of Parthenium. We've taken a long look as to why it is this plant can spread around the globe so easily. It seems like there are a number of cases where the weed is being moved directly from the Americas to countries like India and Australia, Israel, Egypt, by us importing seeds from those countries. But many other cases are simply because they are close to the borders of the country that has had the weed. So being adjacent to those countries, the introduction of the weed has occurred by travel across those borders, perhaps attached to vehicles, perhaps attached to goods that are being imported from those adjacent countries. So a very large number of countries have received the weed by this mechanism. Of course, there's some countries we have no idea how the weed got there. And there are some countries where we think that military activities took the weed to those countries. So it's very interesting by analyzing these different routes of spread, how it might be that we can prevent further spread in the future. This map just quickly summarizes what's occurred in the South Indian Ocean, South African region. The first populations were found in Mauritius, and perhaps a more dominant population then appeared in Madagascar in 1931. And from there, perhaps it then invaded South Africa. And more recently, of course, the weed is spread into Swaziland, Botswana, and Zimbabwe. So once the introduction has occurred, you know, these secondary spreads are really quite common. This is the situation in Southeast Asia. We think the very first populations may have come into northern Vietnam in the 1920s, possibly again through military action or the movement of the French army in that particular country. And very quickly it spread into Yunnan in China and Guangdong in China. And since that time, it's spread to a large number of provinces within China. It got into Japan by an unknown mechanism in the 1970s. And since then, it's got into Thailand very recently, Malaysia fairly recently. And very interestingly, there's been a new introduction. It's genetically different to the southern Chinese populations, an introduction into the port near Shandong in the northern part of China. Perhaps this one came from Korea. We just really don't know. But here is the history of introduction into South Asia. Interestingly enough, there was a small population of Parthenium weed discovered in Kolkata in the 1800s, probably in the Botanic Gardens. We're not sure if it ever spread out of the gardens, possibly not, but that population probably is died out. And the most important introduction then followed in the 1950s into Pune, as we've already heard on several occasions, as a contaminant of wheat. And very rapidly it spread right up to Jammu and Kashmir by the early 1960s, also to Delhi in the 1960s. So its movement was really quite quick, possibly along the railway lines by other roads and pathways in which people were traveling. It got into Nepal, presumably from India, by 1967. And more recently, it spread into Pakistan in the 1980s, Bhutan in the 1980s, and Bangladesh, it's reported in Rajshahi in 1988, a border province of Bangladesh. And since then, it spread fairly widely in Bangladesh. And in 1999, it was introduced into Sri Lanka. So the weed has dramatically spread after major introductions, the major introduction probably being the one into Pune. And from there, we've seen it spread into many other countries. One of the obvious ways it spreads is in markets like this. This is a market in Ethiopia. Many of these seed samples being sold in the market contain Parthenium weed seeds, and consequently, those seeds are taken back and in some cases planted. So the weed can spread very easily within a country by the movement of these kinds of seed lots. And of course, also on vehicles. So these are the major methods which the weed spreads once it gets introduced into a country. And just to highlight how quickly this can be, we undertook a study in Pakistan. It was done by Asad Shabir back in the year 2000 when he did his MPhil study. And the only populations of Parthenium he could find were around Lahore and around Islamabad. And then he did the same study yet again in 2010, using the same techniques, going to the same areas and further afield. And this is the pattern he found. So just within that 10-year period, the weed had dramatically spread. And interestingly enough, if you look closely at the data set, you can see that the route of spread is along the major roadways. So this is where we believe that transport was one of the major vectors for spread. So these are the kinds of global situation we find ourselves in. The plant is spread dramatically from the 1700s right through to the present day. And the problems the plant is causing are also growing. Let me just talk you through a few of these, because maybe you wouldn't have seen some of the problems this plant is causing in other countries. This is Ethiopia. This is a land where this gentleman is trying to graze his cattle. All you can see is Parthenium weed. These cattle are actually eating the rosettes of the Parthenium, which are down close to the ground. That's all they have to eat. This gentleman told us that the cattle, when milked, produced a milk that was very sour. And so clearly the Parthenium had been affecting their milk production. This is a grassland in Australia. You can see the population probably started along that fence line and since then has spread out across the grassland. So again, this is a beef production area, and so dramatic reduction in beef production in such a landscape. And we've had some figures put on the amount of loss to the beef production in Australia. In the early years, it was estimated to be around $14 million Australian. More recently, it's been predicted to be closer to $120 million per annum. So that's a vast amount of money, and that is only on beef production. Of course, it's not only in the pastures and rangelands this weed is to be found. This is a crop of sorghum in Ethiopia, and you can see that it's heavily dominated by Parthenium weed and other Asteraceae weeds. This is a crop of peanuts and maize in China, again, heavily invaded by Parthenium. And this is some aerobic rice in West Bengal. Dr. Ghosh sent me this photograph showing the extent of Parthenium in that particular crop. This is a small crop of vegetables in Bangladesh, onions. The weed is to be found in all of these small crops as well as onion. And again, some estimates have been put on the crop losses due to Parthenium weed. Some reports have suggested about 40% of the annual wheat production is lost due to Parthenium in certain countries, maize 60%, and so on down the list. So you can see there are significant losses to crop production around the globe. It's very difficult to actually get a meaningful figure for the amount of crop loss that may be occurring. But of course, it's not just our crops and our rangelands, it's also everywhere else we find this weed. Here's a photograph in Kenya taken by the team in South Africa showing the weed growing all along the sides of the road. So presumably this has come about due to the spread of the seed by vehicles moving along the road. And of course, these areas on the sides of the roads are where a lot of moisture collects. So it's hardly surprising that this is one of the regions where the weed grows. And this is a photograph from Vietnam showing it growing along river edges. We know the seeds float. They can float for up to 40 days and remain viable. So along the river edges is another major way of the weed spreading. And of course, into our natural environments. This is a great concern to us. Many of our national parks are now coming threatened by this particular weed. National parks in India, national parks in Nepal, but here in Kruger National Park in South Africa. And another photograph of the Masai Mara around the wildebeest and zebra there is all Parthenium as it is in the foreground. So these national parks are becoming heavily This is a map of the protected areas or some of the protected areas in India, and you can see that these are all under threat from Parthenium. Climatic matching of these areas with the climate that the weed prefers shows all of these areas coming under threat. And just to continue with a few more images, these are children in South Africa going to school. Clearly there's an impact upon their health having to walk so closely through the Parthenium mass. And here again is some homes in South Africa where people are having to actually live in amongst the Parthenium weed. So, you know, this plant is having a major impact on our crops, our pastures, and natural ecosystems. And of course, it also has an impact on our health. So one of the substances in the plant that is explained as being one of the reasons for this health effect is parthenin. It's a sesquiterpene lactone, but there are other sesquiterpene lactones in Parthenium which could also be responsible for the ill health. These chemicals seem to collect in all parts of the plant, but they seem to be most dominant in the very fine hairs on the leaves and in the pollen. And those fine hairs and pollen grains can float over vast distances, up to 8 or 10 kilometers away from the Parthenium population. So in actual fact, you can get allergies from this weed and you may not be within 10 kilometers of it. And they say that there are over 40 million sufferers around the globe every year at any given time with Parthenium allergy. So it's these very fine hairs on the leaves, that the trichome hairs that break off and float in the wind along with the pollen grains that cause these allergies. And the facial dermatitis is one of those major problems that we see. Here are 2 other farmers with the same problem with the contact dermatitis. And apparently it is a miserable disease. The skin becomes very thick. It's also on the face and the neck, the forearms, and not only create itchy skin, but it also creates hay fever and bronchitis. And as we've already heard, there are some reports that it actually can lead to the death of people. Of course, some of these abrasions on the skin can lead to ulcers, and those ulcers can become infected. And so there's secondary problems of the disease as well. So not a very pleasant plant. So you can see why we're very concerned about it. And perhaps by the presentation there, you may have seen some circumstances that you're not presently seeing in India. And certainly there are other countries around the globe where they are not yet seeing the major problems that you have in India. So it's good to share this information as to the extent to which the problem can occur. So let's take a look at management. So we've already heard from Dr. Mishra that early detection or understanding the problems of the weed is very important. So educating the farmers and the land managers about this particular weed and to show them exactly what it is they should be looking out for. So this is a little brochure we produced and is being used in Pakistan to educate the farmers about this particular weed. Now, of course, in Pakistan, the weed has only been there for just over 20 years. So there's a lot of people that are not aware of what it is. So this is quite important that they take this step of early detection. I guess in India, we've gone past this point. There's very little opportunity to detect the weed as it establishes in a new area. But I think for other parts of the globe, this is quite important. Of course, there's some physical techniques we can try. Of course, we'd want to try and incorporate these with other methods, but of course you can use a hoe and remove the weeds. But of course, you need to do this before the seeds set, because what you simply do is lay the plants back onto the ground and the seeds are still viable and they can germinate again. They also use this technique in Vietnam. I've seen them removing Parthenium plants from maize crops. In Ethiopia, they've tried a lot of fire control. It doesn't seem to work in Australia particularly well because the landscapes in which Parthenium are found don't usually have enough fuel to set a good hot fire. But in Ethiopia, out on the flat Ogaden regions, apparently fire seems to work quite well. Now, here's a technique that we use in Australia that I don't think will be particularly helpful in India. We actually wash our vehicles. So vehicles moving from one particular area where there may be a large population of Parthenium, we expect the drivers to go through a washdown facility. And they remove a good proportion of the seed on the vehicle. But we know from studies we've done at the university, this is only a proportion of the seed, but removing some is better than no seed removal at all. We also have the facilities up here, as you see, for the much larger vehicles, the big trucks, the cattle trucks, and they are also washed down in the same way. Of course, this approach requires a lot of water, and some of the systems, of course, can collect the water and recycle it after filtering it. So it is a viable approach, I think, in Australia, but maybe not in many other countries. Now that brings me on to chemical management. Now, we use chemicals in Australia. We tend to do it though only on the sides of the road where the populations have suddenly appeared and we need to get rid of them as quickly as we possibly can because we don't wish to have the seeds spread further. We occasionally walk across some of our landscapes, destroying larger populations of Parthenium with a hand sprayer like this, but it's relatively uncommon. And if we look through the literature, we'll also find some people doing in-crop control of Parthenium. And this is a soybean crop in Uganda, where this gentleman is spraying some form of herbicide. But as we've already heard, you know, Parthenium is quite sensitive to a wide range of herbicides. There are many herbicides that will actually kill it. So we're not short of herbicides to use, but the problems are that as soon as you kill one population with the herbicide, there's another cohort of seedlings appearing. So you have to continually use it, maybe up to 4 or 5 times a year. And of course, the weed can be over vast areas. In Australia, 0.6 of a million hectares are covered. And we certainly are not going to use herbicides over this vast area. So we need other methods of managing the plant. And of course, don't forget the chemicals are also expensive and there is impact on the environment. So they're relatively undesirable in certain circumstances. So we can use herbicides in certain cases when the crop is perhaps a very valuable crop and we can afford the herbicides to manage weeds in that crop, or perhaps along the sides of the roads. But in most other cases, we're not going to use herbicides. So that brings me on to biological control. And there are many forms of insect that can cause damage to Parthenium and rusts, fungi that can cause damage. And we have been deliberately seeking these out in countries where the weed is, where it's a natural home to the weed. And we've looked for agents that are only going to attack Parthenium and nothing else. And biological control is now becoming quite an effective way, or at least part of an effective way to manage Parthenium in a large number of countries. And you can see the countries that have biological control agents either deliberately released or for various reasons the agents have found themselves in those countries. So when you see a photograph like this, it's fairly obvious, I think, why Australia decided to go to biological control. So back in the 1970s, this magic weed had just appeared. Nobody really knew what it was. It was appearing everywhere. It was already over vast areas, so there was little opportunity to use herbicides. So what were we going to do? Australia had been a country where a lot of very successful biological control had been carried out in the 1930s, 40s, and 50s. So surely we could manage this plant by biological control, couldn't we? So that was the idea, at least. And since that time then, Australia has discovered 9 insects and 2 fungi that it's released into the environment in Australia after rigorous testing to make sure that they would attack no other plant apart from Parthenium. And they are having a moderate degree of success. 4 of these agents are quite effective. The others are not quite so effective, but they work as a team. And so any agent can contribute to this management, however small its contribution be. So the very first agent that was released is the Zygogrammer, which I think you will know because it's present in India. It was released in India. We released that in 1980. Very soon after that, Epiblema, a little moth that lays its eggs on the plant and its larvae burrow into the stem of the plant and reduce the amount of photosynthate going to the seeds and therefore reduce the vigor of the plant. And since then, also other agents, Listernotus, which I also feel you might be considering to use in India at some time, Smicronex, a little agent that attacks the seeds, Carmenta, an agent that attacks the roots of the plant. And we also have a couple of rusts that seem to be doing a minor degree of effect in the field. The summer rust, which was the first released, not really having a major effect, but the winter rust seems to be, have I got it the wrong way around? The winter rust was the first release and is not having a great effect, and the summer rust is having more of an effect. So these agents that we've released can attack the plant at a wide number of stages of its growth. We've got agents who attack the roots, the leaves, the stems, the flowers, and the seeds. So you would think with all of those agents doing all of that damage, we would have really good control. But the problem is that not all these agents are present at the same time in the field. They're not all present at the same areas in the field. And so we would only perhaps expect to find one or two of these agents acting together at any given time. The one agent that many people feel is quite effective is the Mexican beetle, Zygogrammer bicolorata. This is an agent that attacks the flowers, the leaves, and the adults do this as well as the larvae. So it is quite an effective agent. And this agent now has been either introduced or found itself into 7, 8 different countries. So it's clearly one of the agents that we rely on, certainly in Australia, and I think you also use it significantly in India. This is what the beetle can do. If you place just half a dozen beetles on a young Parthenium plant like that, come back a few days later, those little beetles will have consumed a lot of the vegetative material, if not all of it, and reduce the capacity of the plant to produce any seeds whatsoever. But these are ideal circumstances. It doesn't always occur like this, but a very effective agent all the same. Here's another example. Now, this particular agent, I don't think is suited to India, because this particular agent, the Epiblema moth, has been shown to attack Niger, an oilseed crop that you grow in India. And unfortunately, well, fortunately for us, we don't grow it in Australia. And this agent has been quite an effective agent in Australia. And one of the ways it works is not necessarily to destroy the whole of the Parthenium plant. What it seems to do is prevent the seed producing, the plant producing viable seeds. So over here is an X-ray of a typical Parthenium plant, plenty of filled seeds. And over here is an Epiblema damaged plant, and there's hardly any filled seeds there at all. So what do we do in Australia? Well, of course, once these agents are released, we do expect them to naturally spread, but we can help that process, and community raising of the biological control agents has been an important part of what we have done in the past. It's not quite so active these days, maybe because the agents have been spread fairly widely, but 20 years ago, we used to build facilities like this in the countryside where we could raise Parthenium plants, raise the Zygogrammer Epiblema on these particular plants, and then take them to the field and release them, give them a little bit of help so they could stay within a small area to begin with, to establish, and then to allow them to spread out from those sites. So this was all done by the local communities. Perhaps there would be one officer who was on salary. The rest was local contribution from the farmers to do this work. Now, interestingly, to summarize biological control, I like using this particular example. Whether it's a good example or not will be up to my colleagues to tell me, but we've been studying a site where biological control has been practiced for a number of years. So in the early years, we went to this site at Mollyamba Creek and we sampled the seed bank at that site. And in those early years, we found a massive seed bank, 30 to 50,000 seeds per square meter. Since that time, the seed bank has declined in size. Now, the Parthenium weed made up 60% or 77% of the total seed bank in those early years. So a massive domination of the seed bank by the weed. Although from year 2000 onwards, Parthenium was present at much lower levels, but still a significant level. Now, what has gone on? Well, clearly something occurred around the year 2000. And what it was that 2 of our biological control agents were found at that site. So we can see from this the kind of damage that the biological control agents can do to the population. But it's important to note that that population never went to zero. Now, we didn't expect it to go to zero, but maybe we hoped it would go a little bit lower than perhaps 8,000 seeds per square meter, because 8,000 seeds per square meter is still a large population. And of course, whenever there's a good rainfall event, that seed bank does grow, seed bank produces a significant population of the weed. So what else occurred? Well, what else occurred was we lost our Parthenium, but we started to gain other important weeds. They were coming in to replace the Parthenium that had been lost. And the native vegetation, which is represented by the blue bars, didn't necessarily go back to the way we wanted it to. So we can see from this, the biological control has done a significant had a significant effect upon the Parthenium weed problem, but on its own, it's not the full solution. So we can summarize what is going on in Australia by some of the comments we read in the literature. Papers report that the Parthenium population has been reduced by 90% in its density, that the weed doesn't grow anywhere near as tall as it used to be. There seems to be a reduction in the flower production, but it does mean we still have a bit of a Parthenium problem. So biological control on its own, as we always expected, is not going to be the full solution. We need other things. And so what we've been doing at the University of Queensland is looking at how our native plants, or perhaps valuable fodder plants, if introduced into the rangeland, could suppress the growth of Parthenium. So we've tested a wide range of plants, and I know this is a technique you guys do in India as well. So in fact, we started doing this work after you did it. But we tested what we regard as quite valuable plants, plants that have a secondary value, in other words, are good fodder plants. And we ran these competition experiments, and we determined from these experiments an index of competition for these plants, or a suppression index. And here's one of the better plants that we discovered. It's not necessarily a good plant, because this plant is also a bit of an environmental weed in Australia, but Buffle Grass was planted into this landscape we see here, heavily dominated by Parthenium, but it suppressed the Parthenium growth. I think you can only see 1 or 2 plants, and by 130 days later, there was no Parthenium left. Of course, there's still seed in the seed bank, so this strategy of planting these suppressive plants would need to be continued in subsequent years. Okay, so we've got a secondary tool. We can use native plants or pasture plants to suppress Parthenium. We can use biological control. And interestingly enough, the two, when used together, do a very good job. So on this side of the graph, we can see a suppressive plant called butterfly pea. You can see as the density of the suppressive plant increases, the number of seeds produced by the Parthenium plant declines, but it doesn't go to zero. But if we add in a biological control agent, the little beetle, the two together can actually suppress seed production almost to zero. So a good example of integrating weed management strategies. So clearly, integrated weed management is the strategy we have to use. I think it will be different in different countries, which components of those that I've shown could be incorporated together, will be different in different countries, and will be different from location to location. So what about the future? I just wanted to say a few words about this, because the strategies we put in place today need to be rigorous into the future. Otherwise, there's little point in using them. So what's going to happen in the future? Well, it's like staring into a crystal ball. It's almost impossible to tell. But of all the different climate change variables that we know of, that is likely to change in a uniform fashion is the concentration of carbon dioxide in the atmosphere. Temperatures, yes, they'll increase, but we don't really know by how much. There'll be changes in rainfall pattern, but these will be different in different regions. And again, it's a little bit difficult to predict what's going to happen in India, in Australia. So we've done most of our work based on elevated CO2, and it gives us some insights into what could occur. So we do all our work in these Convener growth chambers. So we're very fortunate to have these, and we can modify many different things, including temperature, lighting, humidity, and of course, CO2 concentration. And this is one of the very first experiments we did. We grew 2 biotypes of Parthenium, one from ICAR-DWR called T, one from Claremont called C. These plants were grown on this side of the slide under the ambient, the regular CO2, 400 parts per million, which is what we have at the present time, or just a little bit more than that, I guess now. And over on this side, the same plants, grown exactly the same time, so they're the same age. These were grown under 600 parts per million, a concentration we expect to find in the environment by 2050. And I think you can see immediately what the responses of the plants are. So those plants grown under elevated CO2 showed a 200-fold % increase in leaf area. The number of leaves produced was 150% increase. Branches, 50% more branches, and the branch length was greater. The root mass was 200% greater. The shoot biomass was 250% greater. So the total biomass of the plant was about 180% more. And the plants were taller, as we saw in the photograph. They produced 200% more flowers. Their water use efficiency was 2.5-fold greater. That increased transpiration, and their net photosynthesis was greater. So you can see that Parthenium weed is going to dramatically change its ability to grow in the future. And importantly, we discovered those plants grown under the elevated CO2. Now, this is a slightly different experiment. It was run at 550 parts per million, so a little bit less than the previous experiment. And we discovered these plants produced about 66% more seeds. So a fair indication of the growing problem that we face in the future. So how rigorous would our methods of managing this plant be into the future? So you will recall that I talked about using competitive plants to suppress the growth of Parthenium. Well, we grew Parthenium in competition with these competitive plants in our growth chambers. We grew them at the present carbon dioxide concentration and an elevated level. And this particular species, a Soteria species, which is a C4 species, unfortunately, it is less effective in the future. Another C4 species, Estrebola, again, less effective in the future. But a C3 species, this butterfly pea, it seems to be more effective in the future. Now that's because we believe it's a C3 plant. So we've got a C3 Parthenium competing with a C3 competitive plant. And under that relationship, we're getting better suppression. So maybe in amongst our suppressive plants we look for, we need C3 plants. So that's the first take-home message. The second message is that our biological control agents are likely to be just as effective now in the future as they are now. We've done this for a number of the agents. This just shows the effect of this epiblema moth on the biomass of Parthenium plants grown under 400 parts per million without the agent and with the agent. So as we know, the agent suppresses the biomass, so that was not surprising. Under the elevated CO2, we can see that the Parthenium plant increases its biomass dramatically, but we can see the epiblemma actually suppressed quite reasonably the growth. So we're quite confident that this particular agent, along with the Mexican beetle, are likely still to be quite suppressive in the future to the growth of Parthenium. So that's just looking at elevated CO2. Of course, that is only one of a number of things that we expect to occur. Of course, Parthenium is going to be exposed to increased fire and drought and degraded soils due to what is likely to occur in the future. There's the elevated CO2, the global warming at the same time, the frequency of extreme weather patterns and the global warming are all going to affect our Parthenium. And we can only imagine, as we see in a lot of invasive plants, that this is just going to stimulate the invasiveness of them. We expect these disturbances to increase their establishment. We expect these elevated CO2s and warming to increase the availability of seeds, which of course will increase the competition on our crops. The global warming is likely to help spread the range of the weed, increasing it to different altitudes as well. And the extreme weather patterns like flooding, cyclonic activity, etc., is likely to increase the dispersal. So with all that taken into account, I think we are faced with a very difficult problem. And it's not only with Parthenium weed, it's with a lot of our invasive plants. So our land managers need to be aware of this. It's not absolutely clear what they need to do, but they should take it into consideration and start considering what they need to do. Well, let me just close my presentation with a few concluding remarks. So Parthenium, unfortunately, when I started my research career some 30 odd years ago on Parthenium, it was already a problem weed in about 15 countries. Now that's spread to around 40 plus countries in just a very short period of time. It's rapidly spreading around the world, including the native countries. It's found in about 80 countries in total. And we would have to say it's invasive in at least 30 of these countries. Of course, it's going to increase in its invasiveness in other countries with time. It's a weed of crops. It's become a weed of crops. It naturally started as a weed of pastures, but most of our countries now are reporting invasion in the crops. It's a weed of our natural environments. It's an allergenic plant. It causes problems with our animal health, something I didn't mention earlier on, but I'm sure you know. It's allelopathic, and another study we've done has shown its allelopathic capacity will increase under climate change. So that's another negative for the weed. And it is being spread by many, many pathways. So, you know, if we just wash vehicles, it's not going to be enough because it's going to blow in the wind, it's going to float in the water, it's going to travel in seed lots. So we've got to do a lot of things if we're going to cut down on the spread of the plant. So it's becoming a major problem in many countries. It's not just Australia, not just India. There are countries that are almost as badly affected as we are. But I believe the way forward is to build our management around biological control. It's not the full answer. We need to integrate with that other methods of control. I've shown how competitive plants can help. But of course, using chemicals along the edges of the road to clean up those populations, to ensure that their seed lots, when we share them within country, are clean of weed seeds, etc., etc. There's a lot of things we should do. Now, unfortunately, climate change is going to exacerbate this problem. It's going to make the plants bigger, more actively reproductive, and some of their tools are going to be less effective. I didn't talk about the efficacy of herbicides, but it seems like herbicides may be less effective in the future. So integrated weed management built on a platform of biological control, an approach that targets the seed production, I think is quite critical. And if we do that, we may eventually get some control on this particular weed. Just finally, I'd like to mention that we do have a Parthenium weed network, and everybody is invited to join that network. You'll note that we have people around the globe that are part of this network, people in countries where you don't expect Parthenium to be found, but they're on the network because they are preparing for the fact that weed may come to their country. Already, Malaysia early on was a country that was part of our network. They didn't have the weed, nor did Thailand. And it was through our network that they discovered the weed first in those countries, and they acted upon that early discovery. So the network clearly has some value. The network also prepares a newsletter where we share our ideas. And if anybody would like a copy of the newsletter, then just send myself or Asid Shabir an email, and we'd be more than happy to join you up to the network. So with that, thank you very much for this opportunity to talk to you about interesting weed, Parthenium. It's given me a lot of work over the years. I still expect to do a few more years work on it. And certainly the collaborations I've had with the scientists in India have been very great. I've been very grateful for. I think I've traveled to India over 10 times now. And I see many people online today that we've had active collaborations with, and I'm so very grateful for that. So only through sharing information are we able to move forward. So with that, thank you very much.
Thank you. Thank you very much, Dr. Steve. Really, it was a wonderful talk, and you have in detail, you have explained each and everything here. And just what I want to share before the house about my experience on climate change. In fact, at our directorate, we have done some studies and on this Zygogrammer bicolorata, and we found that reproductive efficiency of this Zygogrammer will be reduced, but eating efficiency will increase. And we found the reason that because in search of when there will be under the elevated CO2 and temperature, there will be demand less, there will be less nitrogen in the plant. So in efforts to get more nitrogen, the eating efficiency of this Biogens was found increased. So we see that, of course, reproductive efficiency is being decreased, but because of increasing efficiency of eating, the almost the situation will be like equivalent as it is as now. So yes. With this remark, really very much thank you, and you have given a very good time to us. And now I invite the participants. Now the talk is open for discussion. So if somebody wants to have some questions with Dr. Steve, I think he will be very much glad to give.
Dr. Sushil Kumar, yeah. We can request.
Oh yeah, you have come, sir.
To unshare the screen.
Yeah. Okay,
that is one thing. I must congratulate and compliment for giving a wonderful talk, particularly talking on the problems of Parthenium at global level. And he has also touched many aspects of Parthenium and its research, what kind of research is going on in different countries, including Australia also. So we are all enriched with the knowledge what he has shared today. My only one query for Dr. Adkins, Steve Adkins. He has shown some of the slides impact of elevated carbon dioxide on weed like Parthenium, what will happen to it in the elevated carbon dioxide. But as far as our knowledge or understanding goes, it is not the only carbon dioxide is increasing. Concomitantly, temperature is also increasing. Therefore, what is the combined impact of elevated temperature and carbon dioxide in this decade? I want to know from the Professor Atkins.
Yeah. Well, we have done some experiments in this area. And the warmer temperatures, unless we go to very high temperatures, also increase the growth of Parthenium. So the two are synergistic up to about 35 degrees centigrade. As we get warmer than that, then the high temperatures begin to have a negative impact on the growth of the plant. But as you rightly point out, these experiments that we do that look at one or perhaps two variables are only giving an indication of what might happen. As Sushil has just pointed out, there are other factors we have to take into account as well. So with the case of the Zygogrammer, we've just simply been looking at the impact on the plant. What's going to happen to the life cycle of the insect under elevated CO2 and elevated temperature? That's also going to have a major effect, not only the nutrition of the plant. So it's really quite complicated. And I guess what we're trying to do here is just allude to the idea that maybe the problem is going to get worse in the future, and we should be prepared for that. There's no scenario in which the plant is going to become less invasive in the future. All the scenarios suggest it's going to become more invasive. And I think that's the message we're trying to get over.
Thank you very much. Dr. Mishra, if you allow me to leave, I will leave now because I have to join some other meetings or people are waiting there.
Yes, sir. Thank you so much, sir. Thank you, sir.
Okay, go ahead. All the best.
Thank you, sir.
Thank you, sir. Thank you. Now, may I invite Dr. N.T. Yaduraju, sir, for any comments?
Thank you, Sushil, for inviting me. And at the outset, I would like to compliment Dr. Stephen Adkins for the excellent presentation. Steve, you have done a wonderful job. The presentation was excellent. And I think the ISWS and the DWR wouldn't have found a better person than you to give a keynote address on Parthenium to inaugurate the Parthenium Awareness Week. And I think I'm very happy that you gave an excellent presentation. I'm sure that the participants will be very much benefited by your presentation and by your observations on what will be the future of Parthenium in the changed climatic scenario. So in all my discussions on Parthenium, I always refer to the example of Australia. We almost got Parthenium in the same time, almost 50 to 55 and 58, but you have been able to contain it to only one state in Queensland, and that is largely because of your rigorous quarantine and emphasis on multiple biocontrol agents, and then your basic study on the soil seed bank and things like that. And you are a pioneer in this area, and I think a lot of credit for managing the Parthenium in Australia should go to you and your team. So I'm not telling just to pep you up, but it's a reality. And as you rightly told, some of the things we may follow, but some of the things we cannot. And now it has become a household name, so there is no point in having a local quarantine of in any area, because it is available throughout the country, and I think it has become a household name. But there are opportunities, and as a chairman of the Research Advisory Committee of the Directorate of Weed Research, Jabalpur, we have been discussing about introducing other biocontrol agents that you have tried with and which are successful in Australia. I hope that in maybe 1 year or maybe maximum 2 years time, I think we will be geared up to introduce many more, at least a couple of more biocontrol agents to take care of this menace. And I think your last few slides were very scary in the sense with increase in carbon dioxide concentration. I think worse is going to come. So I think we must also be prepared for this scenario. And another thing I want to point out is that you are right that we should concentrate on integrated weed management, particularly involving competitive crops and biocontrol agents. And we have been very successful in multiplying the biocontrol agents and then sending it to different parts of the country. And I think Sushil will watch with me, and so also Dr. Mishra, that I think it has been sent to almost every corner of the country. And in some places they are established, in some places it is not. But our experience has been that on its own, the biocontrol is not very satisfactory, not giving satisfactory control.
Yeah.
And I think we have to now think of integrating with, as you rightly pointed out, the competitive crops. And at one time, with efforts of Dr. Mahadevappa, who just recently passed away, we did a lot of work on the cassia, serratia and cassia tora and all those things. And I'm happy that you acknowledge the work of us. But you took over, and then you have intensified the research, but we have lagged behind. So my suggestion to the future, researchers and to the DWR is that we should think of combining the competitive crops along with the idea is very good, and I think that will be a complementary effect you will have. So I think that's one area we can think of, and I'm happy that you also acknowledge the work that DWR is doing with respect to creating awareness. And I copied it from you, Australia, because you are observing Parthenium MMS Day, and I simply copied it when I was the director there. And I'm happy that now it has been extended to a Parthenium Awareness Week, and a lot of awareness work is going on with publications and social media and things like that. But I tell you honestly that this is an important one, but it is not doing much, because in a country, you must have heard about how people are not caring for a mask during the COVID pandemic, not wearing helmets when you are riding the two-wheelers, when they are not worried about the life of their own. I don't think they'll be worried about a small plant like Parthenium. They do not know the impact of Parthenium on biodiversity, environment, and agriculture. But all said and done, it cannot be dropped out. But what I'm telling is we need to do more before it has already become a big problem, but more problem it can come. So I think if we take actions right now, the worst scenario that you are projecting can be minimized. So with this, I would like to thank the Indian Society of Weed Science and the DWR for organizing this particular presentation by you. We are very much benefited. I thank you very much for your presentation and for your being here. Thank you, Steve.
Thank you, sir. Thank you very much for your comments as well as to share your experience about the Parthenium. Yes, I do agree that we have to do a lot if we want to fight against the Parthenium. Not only a single bioagent will not work, certainly. We have to import more bioagents, and I see among us. Meanwhile, I will request the participant that if they are having any specific questions, they can post the questions in the chat box. So we will try to take important questions. But I see that among us, Dr. Gogoi sir is here. Dr. Gogoi had been our in Indian Council of Agriculture Research. He was Assistant Director General. So I invite sir for your quick comments. Dr. Gogoi sir.
Thank you very much, Dr. Sushil Kumar. So along with Dr. Yadira's, I was also working at Jabalpur, sir. We initiated some work with biology, and Sushil was trying to go for biocontrol. So I do agree, we have focused whole global scenario in the context of Parthenium. Only two queries I'd like to know from your vast experience. So number one, so along with suicide gases in the climate change scenarios, so pollen bursting is could be one of the major parameter, as you have mentioned. Now, population dynamics of pollens in the air, when you talked about clean environment. So certainly, the measurement of that threshold level in certain areas which are related to the health hazards, it is, in my opinion, seems to be one of the major issues. So number two, as you know, we had a wildlife sanctuaries. Wildlife sanctuaries we had. Now this particular weed devastating our most of the grasslands in tropics and high humid areas. So in that case, it is creating a major havoc to the wildlife. In these two aspects, can you highlight some your experiences in this regard? Thank you.
It seems as if the weed is now entering many of our protected areas and causing problems to the wildlife. Not only because it is suppressing their natural food source, but also because it's causing ill health to those animals. Although there's little direct evidence of ill health in a wild population of animals. It's been claimed that, let's say that in South Africa that a rhinoceros has been affected by Parthenium weed, but how on earth do you know that for sure? You can't capture the animal, you can't undertake tests. It may not be looking too healthy, but that may be because of other reasons. But we can just assume that these sort of interactions are going on. And so we have to do something about it as soon as we can. They're protected areas. We need to protect them. So consequently, some of the tools that we are developing for agricultural circumstances should also be useful in these protected areas. Clearly, we don't want to introduce non-native plants. This is not a desirable thing to do. So we might be needing to look at the plants that are present in those environments that we believe are more suppressive of the growth of the weed than others, and perhaps more widely spreading those. And using sort of restoration approaches to remove the weed. So in land restoration projects here in Australia, there's a number of different techniques that are used, sort of starting at the center of the weed infestation and gradually moving out, getting rid of the core problem and then mopping up the outliers. So for environmental control, I would assume this is the sort of thing we need to do, but there's so little research that's been undertaken on how we might do that.
Thank you, sir. Really, Gagoi sir, you have raised a very valid point that Parthenium is increasing in protective lands. In fact, earlier it was not a problem in India in forest areas, but now about 2 million hectares area of the forest is invaded by the weed. And every year we are receiving many complaints from the forest officers and managers about the weed. But unfortunately, just I want to bring in the knowledge of the House that in protective areas, there are restrictions that they cannot release any exotic things. So we are planning to have a dialogue with the forest officers, with the Ministry of the Forest and Environment, whether we should, therefore they should allow the introduction of the weeds, that Biogens. So here, any other questions people may, Dr. R.M. Kathiresan is here. And before I invite you, sir, may I invite Dr. Samundar Singh here for his comments. Dr. Samundar is the president of International Weed Science Society and very knowledgeable person. So Dr. Samundar Singh, please.
Thank you, Sushil. Thank you, Steve's excellent presentation. You missed one point, that was agronomic manipulations. You know, we are agronomists. And the important thing in this case is, you know, the depth of emergence of, you know, Parthenium. Okay. Dr. Yaduraju said we can, you know, go for crop rotation. That's not easy in a country like India. So what we have found that if you bury the seed more than 4 centimeters, it won't emerge.
Yep.
Okay.
Yep.
Secondly, like say we have rice cultivation also. So when there is flooding of water, again that can reduce the emergence of Parthenium.
Yep.
when we talk about environment, many a time we feel that, it's going down, but this is all because of, the weather pattern. Whenever there is rain, in case of, March or April, there is a huge flush of Parthenium. But if there's no rain that time, its population goes down. So this is a major effect we have in case of North India. If you feel, I can share the screen. Anyway, it's not coming, but we have done some work in this case. And we found that, seeding depth has significant effect on emergence. I can share you the photo with you later on. And the same is for flooding also.
Yep.
We tried to do some comparative studies with pearl millet, but we didn't found, a good effect of that also. So if you look practically, it's coming in all crop, whether this is sorghum, maize, or pearl millet. But yes, Susbania can express it, but not many other crops. So that is, what we have been doing here in ISAR.
Right.
Thank you very much again.
Thank you, Dr. Samundar Singh. Now, may I invite Dr. Kathiresan sir? Dr. Kathiresan sir, yeah, please. Please unmute.
Yeah, could you hear me?
Yeah.
Yeah, thank you, Dr. Sushil, and thank you, Dr. Steve. It's a nice presentation, wonderful presentation. I have been in association with Dr. Steve for quite long, and I have only 2 points, and those 2 points have already been pointed out. But of course, we have done some work, and that is with climate change, with increasing increasing temperature and reducing soil moisture. Both are combined. As you said, we have seen that the germinability of Parthenium seeds comes down when the temperature goes above 35 degrees Celsius, which is quite often experienced in the South Indian conditions. And that increase in temperature is always coupled with reducing soil moisture. So for Parthenium germination, what we have observed is combined factor of soil moisture above 80% and temperature below 30 to between 30 and 35 is optimum. So with global warming, the soil temperature in conditions like India may go above 35, and at the same time, the soil moisture will recede below 80. So that may have an adverse effect on the germination of Parthenium. That is one thing. And I would like to know about your suggestions regarding that. And number 2 is Dr. Bhaskar, ADG, was initially telling that harrowing it in young stages of Parthenium will help management. And in that context, I have one observation. You are mentioning about that sesquiterpene lactone parthenin in Parthenium, and that sesquiterpene lactone parthenin is autoallelopathic on Parthenium itself. So the young seedlings, when they are either decomposed or ploughed in, that sesquiterpene lactone is injurious over the same Parthenium plant, and it is more effective when the young plants are harrowed. So I would like to know about your suggestions in these 2 issues.
Yeah. Okay. Well, it's good to see you again, RM.
Yeah.
It's been a few years. I mean, clearly there's a lot to consider as to what will happen to the life cycle of Parthenium as we move into a climate change circumstance. So as you rightly say, as the temperatures and the moisture gets hot and the temperatures drop down, the number of plants that germinate may become a lot less. But that's just in one particular area. That may not apply to other areas. So maybe you'll be a little luckier, whereas others are not going to be so lucky. And you rightly say that, you know, when you harrow Parthenium into the soil, it's going to release these sesquiterpene lactones, and they are inhibitory to the growth of other plants. To some degree, they're inhibitory to Parthenium itself, but in fact, they are more inhibitory to other plants. So, and what we need to know is the length of time that they remain active in the soil. And some early studies have shown that Parthenium, one of the major sesquiterpene lactones, has a very short life in the soil. and breaks down quite quickly. And because of that argument, some people have suggested it's not the major allelopathic substance in the plant, but that's a separate story. So yeah, by harrowing in the young plants, you're going to get a seed bed that is a little less likely to support the growth of crop seeds, but it may also prevent the germination and establishment of Parthenium as well. So we need to try it to work out exactly what it would do.
Thank you. Thank you.
Thank you.
So in chat box, I find that people are interested to know, Dr. Steve, that how community raising of Biogens is being used for control of Parthenium in Australia. You might have talked about that, no? So
yes, that's right.
Yeah.
So clearly you can collect the agents from the field. So once the agents have been released, they're there in the field, but they may not be there at high enough densities to do a lot of damage, or maybe they're not in every particular location where you would like to see them. So you can go to the field and you can collect some of these agents, and then you can increase the numbers by growing them in these community settings. And then you can release the larger number of the beetles back into the environment if there's a Mexican beetle you're raising. Mexican beetles seem to be fairly okay. Some of the other agents, no, they're not easy to raise at all.
Okay, thank you. Thank you, sir. I find Dr. Asad Sabir here. Dr. Sabir, will you comment anything? Yeah. Yes, please.
Yes. Thank you, Dr. Sushil, for giving me opportunity. It's great to see you all here. Sorry, my daughter is making bit.
No problem, please. Please go ahead.
Yeah, so it's great to see you all, and such a great talk, Steve. As always is the case. It's great to see that you provided an overview of Parthenium weed issue around the world, and discuss the various approaches being used in Australia and how these can be implemented in other parts of the world. Yes, I think that's great.
Well, a lot of what I presented was your work, so we should be very grateful.
In fact, for the information of house, I want to bring into the notice of them that Dr. Asid Sabir and Dr. Steve, they have worked nicely together in the lab of Australia, and Dr. Sabir is also doing great work in Pakistan on Parthenium. So thank you very much, Dr. Sabir. And may I have any comments? Some people I find from Bangladesh. If you want to share your experience, Dr. Arif, I saw your name there in the chat box. Okay, if he is not there, I invite any other person.
Sushil.
Yeah, Dr. Chittamathu. Yeah, please.
Yeah. This is for Steve. Dr. Steve, we from Tamil Nadu in TNA, Tamil Nadu Agriculture University.
Yes.
We have tried with different herbicides like 2,4-D, glyphosate, paraquat. If you go with the paraquat, it did not control the Parthenium, even at the young stage as well as at the flowering stage. Though we are calling or saying that paraquat is a total contact herbicides, photoactive herbicides, even under bright sunshine, it could not control effectively the Parthenium. What may be the reason behind it? Though it is a contact herbicide, it's not able to control the Parthenium. May I have any remarks on this issue?
Well, herbicides are not my area of expertise. As you say, paraquat should control Parthenium. If the plants are growing under stressful environments, as you would probably know, the plant doesn't take up the herbicide so easily, and then you don't get such good control. But paraquat is not a translocatable herbicide, it's a contact herbicide. So you, even under stressful conditions, you should get some degree of control. I really don't know. I mean, it's a plant that has fairly hairy leaves and they tend to prevent the uptake of herbicides. But maybe there's others online that could give you a better answer than I can. Unfortunately, it's not something I know very well. It's been reported that paraquat does control Parthenium, so maybe the environmental circumstances in which the plants were growing, some component.
We have tried in the open field. I controlled all even Datura and grasses and other broadleaf feeds, but except Parthenium, it's left as such without killing. I will share the pictures with me after some times. Okay. It's amazing to see that it remain green and fleshy. Yeah.
Okay, thank you, Dr. Chinnamuthu, for your inquiry as well as your comments. Anybody else who wants to put any question to Dr. Steve? Yeah, please. Please, Doctor, go ahead, Dr. Rulandi.
Yeah, I am Dr. Irlandi. Presently, I am a Indian Forest Service officer retired. I am the old alumni of the IRA. Basically, I am an entomologist.
Yeah, good.
In fact, I widely travel in Australia during my service. To be specific, I have visited the Brisbane, the land also. As rightly Dr. Stephen was pointing out, there's a wide spread of Parthenium I could see during our travel in all the areas on Brisbane. Coming to this one, this seed formation, of course, I put it in the chart box. Whether the seed formation, I mean, the season varies from country to country. The season.
The seasons?
Yeah.
Yeah, look, we have quite pronounced seasons in the regions where Parthenium grows in Australia. Yeah. So for a large portion of the year, our winter months, the plant is not present in the field as a plant. Of course, it's in the seed bank as a seed. And then when the spring months come around, like in about 2 or 3 more months' time, germination will occur and populations are present over the summer months.
Okay. My second, yeah, please.
So it's different to what you see in India, where you can actually have the plant present in the field throughout the whole of the year.
Okay. My second question is, I mean, this one clarification is, at what stage these the biocontrol agents, to be specific, the Mexican beetles are being, I mean, taken for the biocontrol purpose from the community-raising the centers?
Sorry, I dropped out for a few moments there. So I think you're asking about the biological control agents and how they survive through the winter. Well, maybe Dr. Shabir could answer that one better than I can, but I understand the beetle goes into a diapause and is present in the soil around the plants, as do most of the other agents. So there's a resting phase that occurs during that period of time. Of course, the rusts remain in the field as spores. So they persist, but by what mechanism is different depending on the agent. But certainly the Mexican beetle diapauses and is found in the ground close to where the plants were. And they hatch. And this is, one of the reasons why we may not get good control by one single biological control agent, because the hatching could be at different times in different places. They may not match up perfectly with when the Parthenium is present in the field. But if you've got more agents, then there's a greater chance of getting some degree of control. So I always think that you need something in the order of 3, 4, even 5 agents that are active to get good. For Parthenium weed, you need 4 or 5 agents.
Thank you, Dr. Steve, and thank you, Dr. Irlandi. Now I think we have taken almost 45 minutes more from the scheduled time. And so now I think I will request all the participants, those are having some questions in their mind, they should post their questions in the chat box, and we will certainly forward those questions to Dr. Steve to give the reply in due course. So now we are on the verge of concluding the sessions. Before giving the vote of thanks, may I request our chairman, Dr. A.K. Pandey Sahab, for concluding remarks. Dr. Pandey Sahab. May I request Dr. A.K. Pandey sir for his concluding remarks. I'm not getting any response from him. So I'm now on the behalf of Indian Society of Weed Science and Directorate of Weed Research, I am really very much thankful to Dr. Steve. First of all, Dr. Steve, because he has for his keynote address, and he has really enriched our knowledge about the Parthenium and about the management tactics, and we have certainly learned that how we can replicate the management methods what is being done in Australia. Certainly the primitive method as is being done in Australia is not possible, I think, in the case of India. But in spite of that, sir, we will try to follow, we will try to introduce more insects for biological control, and we will try to integrate other methods also. Thank you very much for your nice talk. And I'm really very much thankful to our Director General, Dr. Trilochan Mahapatra. When we were planning Parthenium Awareness Week, we requested him to give an appeal so that that appeal could be given to all the Krishi Vigyan Kendras and state agriculture universities and to the ICR institutes. You know that there are more than 100 ICR institutes with their hundreds of regional stations. So by this way, as indicated by Director DWR, Dr. J.S. Mishra, that about 10,000 to 10,000 stakeholders, we have given that message. So I'm really very much thankful to our Director General for sparing his time to write the appeal and pass on to us for giving to the stakeholders. I am very much thankful to our DDG Sahab, Dr. S.K. Chaudhary. You know that in spite of his very busy schedules, and he was having another very important meeting called by the ministers, but still he spared his time, valuable time, to record the message, and he gave his address online. And really, sir, we are very much thankful to him for his nice address. We are very much thankful to our ADG Sahab, Dr. Bhaskar Sahab also, and who was attending this webinar till the end, about I think about up to 2 o'clock, and he gave a very he shared his experience very nicely, and that how Parthenium can be managed in India. We are very much thankful to Dr. Bhaskar Sahab. We are very much thankful to our chairman, Dr. A.K. Pandey Sahab. Really, he has given a lot of time to us. And in fact, I want to bring to the knowledge of how that he has spread this message to the whole Ujjain area, and now collectorates, offices, agriculture offices, etc. He has passed on that message, and he is doing some activities during this Parthenium Awareness Week. He has also given the message to his all the colleges affiliated with the Ujjain University to do some activities on that Parthenium Awareness Week. We are very much thankful to him. We are very much thankful to our stalwarts and our senior colleagues, Dr. Yaduraju sir, Dr. Gogoi sir, Dr. Kathy Resan Saab, Dr. Samundar Singh sir, and many others. Dr. VP Singh, I am seeing, is also there. So Dr. Chinnamuthu, our Vice President, Dr. Chinnamuthu, Dr. T.K. Das, and moreover, I am really very much thankful to all the participants. I want to bring to your kind knowledge that when Dr. Steve was giving his keynote address, then about 478 people were participating, listening his talk. So I am very much thankful to all of the participants, those who have taken, those who have participated in this talk. And at the end, I am very much thankful to my own colleagues from DWR who are taking part in with very vigorous and zeal in this Parthenium Awareness Week. I am very much thankful to Mr. Gyanendra Pratap Singh, who is just dealing with this computer and IT sections also for their participation, as well as dealing with the system. So thank you very much at the end. Thank you very much to all of you again. Thank you very much, sir. And with this, I take the permission from the chairman and other seniors to close the webinar. Thank you very much, Steve, again, and thank you very much to all. And before that,
thank you very much.
Okay, thank you, sir. Thank you.
Thank you very much.
Thank you, Steve. Thank you, Steve.
Thank you, Dr. Asad. So now I will request
thank you.
Gyanendra to